Hello and welcome to the Beyond Biotech podcast number 72. I'm Jim Cornell and this is the weekly Labiotech podcast. And this week we have two guests from the same company, and that company is Phenotypeca, based in Nottingham in the UK. The company does work in a lot of different areas, which we will hear about later, but the main focus today is the company's partnership with the Bill and Melinda Gates Foundation on reducing the cost of vaccines in low and middle income countries. So to tell us about that partnership in reducing the cost of vaccines, as well as its other work, and the origins of Phenotypeca, are Research and Development Director, Professor Ed Louie, and the CEO, Johnny Cordiner, who you will hear from first.
The company is really based upon two scientific backgrounds. One was Chris McFinnis, and the other one is Ed, who's on the podcast with us. And they identified a fusion of two technologies, which I'll let Ed explain a little bit more in detail in a second. But as a consequence of the fusion of those two things, it gave a dexterity to be able to bring to the manufacture of recombinant proteins a greater ability to use the full power of biology. And as a consequence of that, you can do something which I think scientists would understand, which is bespoke optimization, genuine multi-parameter optimization. Now, all recombinant protein manufacture does not require that solution, but there are certain commercial and technical spots where the ability to optimize creates a premium product which can command market share and is commercially valuable. And in essence, that's what we do. We carefully scope out where the skills of our technology answer a commercially and technically relevant problem, and we bring it to bear on those cases. And we do two things in parallel. We generate revenue by doing things that are revenue generating, which we'll get into deeper in a second, but we also at the same time create value through product. And our technology as such doesn't create product, but when you get into collaboration with synergistic technologies, the combination of the two can create very powerful and breakthrough product, and that's how we deploy it. So maybe Ed can give you a little bit of background as to the sort of run up to the company and the conversations that led to that.
Yeah. So Chris Finnis, one of the founders, is one of the world experts in the traditional industrial approach to yeast, baker's yeast as a microbial chassis for recombinant protein production. So baker's yeast was the first used for recombinant protein production. Insulin's been on the market made in yeast for over 40 years now, I think. And the way the market worked and the way companies worked in this area is they kept the know-how very secret. So there was only a couple of companies that were successfully making products with yeast, and it closed off most of the market. So this was through traditional means. There was a strain that was reasonably good. Various lessons were learned either from academia or from R&D within the industries of engineering steps that would improve recombinant protein production. For example, there's a well-known protease that was discovered in the 70s that is now incorporated into every production strain and long off patent, so everybody can use it. But also, random mutagenesis was used. So you would mutate your strain, screen for things that were better, use that for production, do more and more rounds of mutagenesis. The problem with that is it's random. You don't know what it is that the improvements are. And because it's mutagenesis, lots of off-target detrimental things occur in your strain. So that is the traditional approach. There was incremental improvements to production over the decades, and it took decades to make the improvements. Chris is an expert in all of these first generation levels. So I came from an academic background. I've been a yeast geneticist molecular geneticist, genomicist for 40 years. I've recently retired from academia, but I was involved in the yeast genome sequencing project, the discovery of new yeast species, and in the last 20 years, the description of the huge amount of genetic diversity that's out there in the world of yeast. So Baker's yeast is found all over the world in various ecological niches. Obviously, it's been a friend to humanity for tens of thousands of years with fermentation products as well as making bread, but it's been limited in the strains that were available for industry up until recently because there was a few lab strains being used. They were all related to each other, generated in academic labs by crossing. And those that behaved well were the ones that were taken up by industries. So industries were limited to a single or a few related strains. Whereas what I've been doing in my group for 20, 25 years was to look at this wide range of genetic diversity from various places. We sequenced many genomes in the old-fashioned way. We measured traits, 201 traits actually, across dozens of strains, showed there was a strong correlation between these phenotypes and the genotypes, which led us to the realization that by classical breeding between these strains, we should be able to determine the underlying genetic variation that underpins the improvement of a trait. So if you were making more ethanol, for example, or less ethanol in a wine or brewing in this situation, you could not only generate strains that do this, but find the underlying genetic variation that can do this. So lots of yeasty traits can be tackled this way, but it also applies to non-yeast things. So if you're trying to express a protein in yeast and get it secreted, that is the level of expression, the amount of secretion, the amount of post-translational modifications. Those are all phenotypic traits, and there is variation in those phenotypic traits when you look at a wider variety of strains. So once I realized that you could very fine detail, very high resolution determine underlying genetic variation underpinning complex traits, I started going around describing this in meetings and was invited to a Novozymes, Novo Nordisk annual tech fest where every R&D group from around the world got together and they would invite one or two external people to talk about the latest things. So this was about 10 years ago, and I was one of the invitees, and Chris was there as he was working for Novozymes at the time. Interestingly enough, or coincidentally, it happened to be in a subsidiary that was here in Nottingham. So he heard the talk there, and a few months later, I got invited to the Nottingham location. At the time, I was at the University of Nottingham, so very close by, and discussed the potential for using breeding in addition to the traditional engineering mutagenesis approach to improve recombinant protein production. Unfortunately, they decided to go their own way. Chris went off on his own, and a few years later came back to me and we rediscussed this, went out and found a number of industries that were willing to partner with us, and we tried to start a spin-out from the University of Leicester, where I was at the time. That didn't work out, but in the meantime, Chris and a business partner founded the company. A few months later, I retired from the university and became the chief scientist, employee number 3, and we now combine the best of the off-patent traditional approach with the breeding technology, which takes advantage of hundreds of thousands of genetic variants that are past natural selection. These are living yeast cells, so they're not detrimental, they're alive, they're robust. And by breeding, we put them in multiple combinations that have never been seen by nature, and always get an expansion of the phenotypic space. So we always get things that are better than the original strains we put in. And not only that, because we can sequence these things, we can identify down to very high resolution what combination of genetic variants on top of the known beneficial engineering give you improved titer of your recombinant protein, robustness to the industrial processes, reduction in downstream processing things, all of these multi-parameter optimizations that you would want. So I'm just going to finish with one thing that I always find interesting that surprises people is that we're dealing with, say, 100,000 genetic variants that we're mixing up together. And that's equivalent to 2 to the 100,000th different genotypes, which each all have their own phenotypic consequences of potential. That's the same as 10 to the 30,000. There are only 10 to the 81 atoms in the universe. So we're essentially talking infinite genetic variation possibilities. And we only sample 10 to the 9th at a time. But 10 to the 9th is enough to identify the combination of 30 different genetic variants that are required for an improvement. And in general, experience shows that 10 variants underpin most traits of interest. So our sampling is enough to get the best strain and identify the combination required to get to the best strain. And that combination that's identified is intellectual property, either as know-how or patents.
Before we get to your technology, could you perhaps briefly explain recombinant protein technology, what it is and how it's applicable?
So the brief definition is expressing from a synthetic piece of DNA on some kind of expression vector or integrated into the genome, a protein that is not found in your chassis, whether it's yeast, E. coli, Chinese hamster ovary cells, insect cells, whatever your chassis is. A recombinant protein is heterologous. It comes from another place. In our case, they're usually human proteins, but it doesn't have to be.
And the commercial context of that is that it's a rapidly expanding field across different industry sectors. In the therapeutic world, then it's already by far the most dominant player in terms of frontline treatments of critical diseases that will be familiar to everybody. But also the largest revenue drugs in the world, the majority of those are recombinant in nature. Monoclonal antibodies, but obviously you've got a burgeoning world of cell and gene therapy, which are not recombinant proteins, but use recombinant proteins in their processing. Insulin would be an example. Monoclonal would be an example. The diseases they treat would be, everything from cancer to osteoarthritis, rheumatoid arthritis, etc., etc. In various industrial sectors, there's a great deal of interest about recombinant proteins, and I guess they're looking for a catalyst that can make that affordable at the right levels. In some cases, that's already the case. In some cases, they need some technology barriers to come down, of which we can be part of that discussion. For instance, in skincare, there's a movement, there's a lot of activity in South Korea at the moment. There's a lot of movement towards peptides and proteins, and they require consistency, quality, and affordability to be able to harness the potential that they bring to skincare products.
As far as vaccines are related to this, what are the issues about supply and affordability currently?
Well, I wouldn't hold this up as being experts in all things vaccine, but in this particular case, albumin is a critical component of mammalian blood, and it has been available for quite some time, but it's derived from animal cells in serum form. That brings with it a number of problems, or can bring problems, not always, but so there's been, for reasons of consistency and reduction of infection risk, etc., there's been a premium for recombinant albumin. But recombinant albumin production in Saccharomyces and baker's yeast has been classically closed out by patents. The patent expired, but the companies involved kept the know-how close to their chest. creating a barrier to entry, they focus on a very high end of the market. So the product is highly priced, and therefore you've got a requirement for in the production, and it's actually present in the final vaccine for albumin, a preference for recombinant albumin, a need for recombinant albumin in certain parts of the world where, for ethical reasons, for instance, with halal, you can't have it from an animal source, but a deficit of supply. So our chairman was a big figure in the vaccine world, and he knew some of the key strategic advisors of Gates Foundation. They had identified the need for albumin at affordable levels for low and middle income to catalyse the affordability of particular vaccines, starting with measles, rubella for children. And we're talking a lot of children. I mean, we're talking, the initial scoping exercise was 100 million, all of which would be, or pretty much all of which would be children. So it's a fairly significant impact that they're looking to catalyse affordability in those vaccines by bringing down the cost of this key excipient. Now, I don't think the Gates, we're not privy to their entire strategy, but I think they take a market forces type approach, where what they're looking to do is ensure that supply is more readily available, and therefore the consumers who would be the vaccine manufacturers have got access to more affordable albumin, which would then bring down the cost of the vaccines to whoever pays for them in these countries.
How can you help in improving all of that, I guess, chain?
One of the things when I joined the company that was attractive to it is that it's built into the DNA of the company, that what they're looking to do, as well as do other things that a normal biotech would do in terms of making return for shareholders, etc., is that they're looking to make key medicines more affordable to vast bulks of the population who can't presently afford it. Now, what that means is bringing down the price of certain medicines in Western markets, but also in lower middle income, going up beyond that threshold where, for the foreseeable future, realistically, vast chunks of the population are not going to be able to afford certain medicines. And can we make the difference of making them affordable? Now, we're just a small company in Nottingham. We can't change the world on our own, but what we can do is be a critical catalyst. So if we are making these medicines manufacturer and capable of manufacture at a price point that makes it affordable for those that pay the bills, and that could well be consumers in these various countries around the world, then there is no longer a convenient excuse as to why they're not bought. Because at the moment, they can't be afforded, so there's no conversation to have. I guess what we do is provoke that conversation. That can expand beyond vaccines. So there are a number of therapeutics that are now off patent or approaching being off patent, for which biosimilars can be developed. And we can use the full power of biology to develop those biosimilars, which may be technically otherwise challenging to do, or certainly technically to do it to bring down the cost substantially. That's part of our DNA of what we're trying to do, and bring them down to affordable levels in different parts of the world. How that model will pay out, Jim, I don't know. There's some big forces to play, but the logical model that you could see is that you have manufacturing done regionally, which accords with the cost base of each region, and therefore it's more affordable for that region. But what you don't want is a race to the bottom. So what you want is the best science, the best regulatory manufacturing practices put in place, but our strains embedded in those environments can certainly achieve that. So you can get world-class efficiency, world-class quality. The manufacturing of these is highly skilled, but it's accessible. It's accessible skills. And the building of the plant is specialized, but is accessible, specialized. So therefore, you can see a regional hub type model where you can put in place strains and make things affordable to vast chunks of the world's population that are not presently available. And we see ourselves as being a catalyst to that, because at the moment, I'm not suggesting there's big dark monsters out there in terms of these big companies, but they've all got shareholders to answer to. So they have to behave in a way that's in accordance with that, so they perhaps don't have the freedom to do certain things that we can do with our technology in a more disruptive manner.
just to continue with the vaccine side a little bit, so we're very far along with recombinant albumin with the Gates Foundation partnership, but we're also partners with VaxHub, which is this large UK consortium of academic institutions and industrial partners for developing new vaccines for a variety of diseases. And we don't know what the direction is, but being part of that will expand our involvement or roles in various vaccines, which will be beyond albumin. It'll be, the antigen production that's used for eliciting an immune response, amongst other potential other excipients that are used.
Could you explain a little bit about your QTL technology, what it is and how it works?
Sure. There's actually a little bit of confusion and therefore some education that needs to be done in the wider community. But QTL technology, quantitative trait loci analysis. So if you have a population of genetically diverse individuals and you have different phenotypes, say it's a disease or in the case of yeast, expression of a certain level of recombinant protein, you can associate DNA variants in the genomes with the levels of trait differences that are out there. So I started out as a human population geneticist over 40 years ago, where we didn't have genome sequence, we just had a few markers, and it essentially was trying to map the few markers that were there, right, alleles at those associated with particular diseases. So, the one I worked on mostly was type 1 diabetes and the HLA system, as well as a few other markers. And so it was a statistical association, and from just human populations, this would be now has turned into genome-wide association studies, or GWAS. So QTL technology is similar to GWAS, but here we generate the population through breeding. So we start with a handful of original progenitor parents, and we interbreed them for a number of generations, mixing up the tens to hundreds of thousands of genetic variants they have between them, creating this near infinite variety of genotypes. And then we apply the statistical associations studies to phenotypic traits and this population of diverse individuals. Traditionally, agriculture, say a farmer would be, taking his best milk producing cows and breeding those with a bull that had been a father of better milk producing cows, and you would, shift the next generation population a little bit better each time. And a huge industry of quantitative genetics statistics arose out of that. But there was no genetics in the way that a Mendelian geneticist thinks about there. Whereas what we're doing is every genetic variant is a Mendelian trait. You can follow it, as with Mendel's peas, but there's a lot of them. And together they make traits in their various combinations, both complex and quantitative. So you don't get all tall and all short, you get the huge range of heights that come out in humans or peas or whatever. And so our current level of technology is to apply pretty sophisticated statistical associations to a sample of sequenced individuals that have also been phenotyped, and we get very high resolution mapping of markers that are associated with the improved trait. And it's usually not, it won't be one marker gives you the improvement. It'll be the combination of several markers. And so you need to work that out. And that's only markers associated, but to actually find the causal genetic variation, you actually have to go in and do experiments further in the lab to validate, is it that marker? Is it one nearby? And combinations of them can give you an improvement, whereas any of the individuals on their own wouldn't give you an improvement at all. So they're not predictable in the way that the classical quantitative genetics statistics would tell you. So, the books of 100 years ago gave you predictability, which works great for improved maize, corn production, milk yields, but it doesn't work for some other things that were like horse racing and camel racing, that higher speeds wasn't easily predicted based on this to the same level as expected. So with the yeast variants underpinning the properties that we're looking at for recombinant protein production, they're not predictable from the classical method. So it's a combination of using informed genetics to design the breeding and reasonably sophisticated statistical analyses.
From my perspective, Jim, having worked in complex biology really for the last couple of decades, there's a number of realities that have come out which we can go into in greater depth if you want to. But the beauty of what I saw about this approach is that you're letting evolution do the work. And then you are back reading the reality of how the outcome that you're screening for and looking for has been achieved based upon sophisticated, effectively functional genomics, which allows you then to iterate again, which is still allowing evolution to do the work. So instead of predicting in a forward way, which I think is fraught with danger with genomics and fraught with disappointment and budgets being wasted, you're letting evolution do the work, and then you're figuring out how it's done it.
What are the benefits of this over other technologies?
Well, the first generation I sort of described a bit before. So there was knowledge gleaned from a variety of experiments that say, well, knock this gene out and you'll get improvement. So you engineer that in, or change the expression pattern of some protein that's in the process that helps, some gene in the system that helps the process, and that gives you an improvement. And then mutagenesis on top of that to get incremental changes, which is fraught with difficulties because there will be detrimental side effects. Mutagenesis is random. You'll hit the one you want after you screen, but there'll be 10 or 20 or 30 other changes in the genome which might not be desirable. And then what do you do with those? How do you get rid of those? It's not an easy thing to do. Another limitation of the first generation is that you start with a single genotype and you're moving one or two steps away from that every time. So you're limiting the space that you're looking at. Whereas we're basically saturating the entirety of available genetic space that is out there, which, as I've described, is essentially infinite compared to the number of atoms in the universe. So that's the baker's yeast system. There are other chassis out there that are also first generation. So bacterial systems, E. coli, have been in the mix for decades. And for certain things, it's clearly the chassis of choice. You get very high titers, but you may also have to deal with post-translational modifications that you do or don't want that aren't there, as well as getting rid of toxic elements. There's always an issue with bacterial systems having toxicity. There's CHO cells, Chinese hamster ovary cells, mammalian cells. Those are well suited for proteins that need mammalian glycosylation patterns, but they're expensive. They're not high volume. There's another yeast system, Pichia, which gives you high titers, but very poor quality initially, so a lot of downstream processing required. And then there's some plant cell systems out there as well. All of them are good for their particular purposes. None of the systems, even our second generation system, is good for everything, but we certainly an improvement on the first generation yeast system, and we have several advantages over many of the others. One thing to always keep in mind is it's the net titer of high quality product that you want at the end. And so when you only look at initial titer, which could be higher in other systems, if you have to spend a lot of time and effort cleaning up that product to get to your high quality product at the end, you may have higher expense and end up with less net product than what we can do with our system.
Could you tell me about the partnership with the Bill and Melinda Gates Foundation?
Sure. So quite a while ago, it probably was an accidental meeting of a reasonably high up person at the Gates Foundation, our chairman, who's a vaccinologist formerly from GSK, and one of our founders discussing requirements for vaccines in lower middle income countries. And they came up with the measles rubella vaccine that is essentially unavailable to a large part of the world because of the expense. And the most expensive part was albumin, recombinant albumin. We had already started making it under an IUK grant. It was one of our test proteins, along with a variety of others. They encouraged us to write a grant that would start the process of trying to produce recombinant albumin at a low cost of goods, which was the major driver for the lack of supply, although there was tremendous demand in lower middle income countries. So we wrote the grant, went through several iterations of what we thought we could produce. We had some partners that were good at modeling cost of goods from both the plant side and maintaining that, as well as the actual production side. And we started out, I think we had a 12-month grant to start out with various milestones, which were first increase the titer from what we had, because we had only just started, we had low titer, to have high quality. It's better than the serum-derived human albumin was the goal, and to reduce the cost of goods. So this was a tremendous challenge. As we were going through this, we identified several technical and other challenges along the way, obtained an addendum supplement to this grant to deal with a couple of those challenges, and actually identified further challenges, both to get the cost of goods reduced, as well as reduce some of the unwanted, undesired byproducts that were there, which would have a knock-on effect of reducing the cost of goods, as well as increase the titer. So we've essentially had 3 rounds of funding for them, and we're still coming towards the end of that. We met the quality goal very quickly. I mean, our recombinant albumin is as good as the gold standard that's out there. You just can't tell the difference with the most sophisticated mass spec analysis. The cost of goods we've gotten down. We haven't incorporated all the improvements there, so that's there. So the last thing is just getting the titer just a little bit higher, but we're almost there. And the final thing is to identify a manufacturing partner to take it to the lower middle income countries. We're relying on interactions with the Gates Foundation to identify the appropriate potential partners, which will eventually, I think, as Johnny said, end up being local producers in the various lower middle income countries producing for their own markets. And that's probably the best way forward. And I mean, this will be the first vaccine component with our work with VaxHub, maybe not through the Gates, but other partners. There'll be more potential to bring vaccines and vaccine components to the lower middle income country markets.
What kind of a timeline do you think that will involve in terms of being able to roll this out?
I think this is where it sort of meets commercial reality, really, Jim, because as I said, our role is really to deliver what they wanted to do to catalyze the change. We're a small organization. We can't drive that catalyst of change. So the best way to do this, I believe, is you need a number of different players. So you need the buyers of the vaccine. You require the health organisations that would be responsible for its distribution and delivery into the arms of children. You require the manufacturer of the vaccine, and you require the manufacturer of the albumin. The manufacturer of the albumin has to be attracted to utilise their capacity or build the capacity for that purpose. And that involves blending markets. But the reason for blending markets is to drive down the cost of the albumin that goes into the vaccine as low as possible, by effectively using some of our commercial upside as a mechanism to drive down the cost and reduce the cost of the manufacture that goes to it. So we're doing our part, and we'll do that. But some of the parts that the ultimate customer, etc., can't be decided by us. So that really depends on the powers that be and how quickly they move to get the vaccine manufactured and the health services lined up, and whoever buys these things lined up as well.
There's still a bit of working up to having a viable vaccine. So, I mean, we've provided samples to academic institution that is also supported by Gates that are mixing the vaccine itself with our albumin to see if it works. That's probably 6 to 12 months of just showing that it is a feasible thing. And it's taken us 18 months to get to where we are. It's going to be 12 months from now before it can even go to a manufacturer, probably. Hopefully between now and then, such a partner will be identified.
Our part of the program is not the rate determining step. We can be pretty quick, but in regulated environments, you can only go as fast as the slowest part, and some of those can drag their heels a bit.
Clearly, though, that's not the only thing that you're working on. What other things are you involved in at the moment?
Well, quite a variety, really, because one of the things I'd say is that at the core of what we do is the science, the machinery, the technology, and just recognise peptides and proteins and optimises accordingly. It's a bit ambiguous or ubiquitous as to whether it's a skincare or it's for a particular industrial purpose or whether it's for a therapeutic. For that, it's just a specification on which we work. So our model is to collaborate, and we're creating value in a number of ways. The first thing to say, we generate revenue by, in some cases, solving people's problems. So producing to spec a strain that fits their criteria, and then we license that strain on pre-agreed terms at the end of the contract. And so we have some contracts of that nature ongoing. But the way in which we create value in product is at the moment in 3 areas. One is biosimilars. We've been through the entire formulary to check where our technology can produce something that gives us an edge in that marketplace. There's a number of reasons why that can happen. It could be to do with patents. It could be to do with technical difficulty. It could be with the requirement really for disruptive pricing to drive down the point of particular markets. And so we're carefully building a portfolio of biosimilars based upon that evaluation of the entire formulae of biological therapeutics. Simultaneously, we're sort of closing a partnership in skincare, where we've identified that there's a requirement for recombinant proteins and peptides, which form ingredients that go into the final formulation. So you sell a concentrate of this ingredient, and they have the assays to be able to verify the activity that the skincare companies are looking for. So we've got a program of key peptides or fragments of proteins that are pre-identified as an appetite in the skincare industry for. We also commercializing different applications of albumin. And then we have a series of collaborations that we are working up with platforms, and they are to be announced. They are in quite sort of exciting areas where recombinant proteins, there's a requirement for the ability for the dexterity of optimization to produce recombinant proteins to a particular specification, and they can push back the barriers of the particular application. So without giving too much away, vaccines, topical delivery of known therapeutics is a couple of areas where we're quite advanced in our discussions, but we're not over the finishing line yet. Plus, we do expect with this collaboration, our partner in the collaboration on the skincare side has somebody we know from our past, very, very sophisticated technology, and they have the ability to push back the barriers in skincare and dermatology. And obviously, our ability to make those to spec is an obvious synergy in that space.
It's definitely often talked about, the inability of low and middle income countries to have access to drugs and treatments, so it's good that that's being addressed. Of course, still a long way to go for all drugs to be affordable for everyone in every part of the world, but hopefully we will get there. Don't forget to check out the latest news and articles at Labiotech.eu, and I hope wherever in the world you are, you have a great week ahead. Thanks for listening, and you'll join us again next time for another Beyond Biotech.